And welcome to Lecture 2, the second in our series of 2 for Chapter 10, Cell Reproduction. So in this particular lesson, we will go ahead and lay out the specifics on mitosis, differentiate the events of the S phase, of interphase, describe the phases of mitosis, differentiate cytokinesis in plants and animals, explain the importance of checkpoints in mitosis, and explain the importance of P53. So we're talking about the eukaryotic cell cycle. Now, like cell division, binary fission, and prokaryotes, eukaryotes must accurately duplicate their genetic material, divide the contents equally in order to effectively form two viable daughter cells. But of course, eukaryotic organisms are more complex than bacteria, so the process of cell division is obviously going to be more complex. Eukaryotic cell cycle is divided into 5 phases, and you can differentiate each of the 5 phases by very specific events that occur in each phase. So you can tell them apart fairly easily by very specific events that go on there. And we'll talk about those as we get into the stages of mitosis. So the 5 phases of the cell cycle are these. So we have the gap or growth phase. The first gap or growth phase, we call it G sub 1 or G1. And this is where the primary growth of the cell takes place. It's the longest in the whole cycle. In other words, it takes the most time. The S phase is the synthesis phase, and you can guess from its name, I'm sure, that this is the period of time when the cell synthesizes or replicates its genome, its DNA. And then we have another gap phase or growth phase. We call it G sub 2 or G2. And that's when the cell is preparing for division, preparing for mitosis. Now, these 3 phases are what we call collectively interphase. Interphase occurs between mitotic divisions, okay? So mitosis then is where the cell actively divides. Spindles, we'll talk about those, attached to sister chromatids. You know what those are already. Chromatids are separated to opposite sides or opposite poles of the parent cell, and then cytokinesis refers to the movement, that's the kinesis part of the cytoplasm, that's the cyto part. So it's division of the cytoplasm into 2 roughly equal portions so that each new daughter cell gets a full complement of cytoplasm. Okay, so diagrammatically the cell cycle looks like this. It'd be a good idea to kind of commit this figure to memory. You have one very similar in your textbook. So here you have the gap 1, growth phase 1, the synthesis phase, that's where DNA replication is occurring, and the gap 2 or growth 2 phase. These 3 phases, we refer to them as interphase. In other words, that's the portion of the cell cycle that occurs between cell divisions, between mitosis. And then in mitosis, we call that the M phase for short, is divided into 5 phases. We'll look at them individually. That's in this lavender color. And then you see in this blue, in the blue arrow, that's cytokinesis. That's where the cytoplasm is divided and the daughter cells, the 2 brand new daughter cells, are formally separated. Okay, so it takes time for the cell cycle to occur, and it varies between organisms and species. Our old friend the fruit fly takes about 8 minutes for division to occur. Mature cells take longer to grow, and so the typical mammalian cell, including humans, takes about 24 hours. Look at that, liver cells take more than a year. Interestingly, growth occurs during the G1, G2, and S phases. Okay, that's why we call them growth 1, growth 2, and in synthesis the cell is still growing because the cell is duplicating, right? It's DNA, so it has to get bigger in order to hold all of that DNA. Most variation occurs in the most variation in length occurs in the growth 1 phase, and the resting phase, G sub 0, or the G0 phase. Here's where the cell sort of takes time off from actively dividing and becomes inactive. This is it says non-cycling state. It's G1. We call it the gap 0 phase, and that's when some cells, not all, but some cells like muscle cells and nerve cells go into a period we call it quiescence, where they essentially don't do anything, because heart muscle cells, cardiac muscle cells, and nerve cells cannot reproduce themselves. So once they divide, right, they can't regenerate themselves. So once they divide, they're done. We'll talk more about that later. Okay, the G0 phase, called the quiescence phase, and you can see here is your little simple diagram of the cell cycle, right? Mitosis is where the cell divides. There's the gap 1, there's synthesis, the S phase, and there's the gap 2. Some cells after division jump out of gap 1, jump out of the growth phase 1 and go into G0. We call that gap 0 or G0. Now some cells after a period of quiescence can jump back into the cycle and continue and divide again. But many cells, like nerve cells and cardiac muscle cells, remain in quiescence. Okay, so some cells are not meant to keep dividing, like heart muscle cells and neurons, and so they remain in quiescence. And like liver and kidney cells, some cells enter a period of extended quiescence, but then they can jump out. It's like hibernation, right? And then they can jump out and go right back into the cycle. All right, so this video is from Professor Dave. It's really quite good and really quite simple, presenting to you mitosis, how one cell becomes two. The link is here, and the actual video is embedded in the notes. Okay, so interphase. Interphase is the phase between intermitotic divisions, all right? And as we've already described, as I've already mentioned to you, it consists of the growth 1, G1 synthesis, and growth 2 phases. In the growth 1 phase, or gap 1 phase, the cell undergoes a major portion of growth. This is the period that takes the longest, or I should say the cell is in G1 longer than any other phase. S is when DNA is replicated, and the gap 2 or growth 2 chromosomes coil around the motor proteins, centrioles are replicated, and here the cell is getting ready to go into active division. Now, you know from your look at the in the first lecture, the structure of a chromosome. That's a centromere is that point of constriction where the two sister chromatids come together, and then they are held together by cohesion proteins. Chromatids stay attached by cohesion. That's cohesion proteins. Okay. And also, I think I mentioned this before, but each sister chromatid more towards the outside, right? The centromere is towards the inside, between the two, more towards the outside of the of each chromatid now is a kinetochore. Kineto comes from kinesis, which comes from movement, and so this is where your microtubules, your spindle apparatus, will attach to the kinetochore during anaphase, and that's how the chromatids get pulled apart, right? They are in motion, they get pulled apart, they're in kinesis. That's the job of the kinetochore. Okay, so let's talk about the phases individually. Here's interphase. This is just a micrograph over here. Here is your illustration. Now in interphase, the cell is growing, right, in G1 and growing in G2 and synthesizing DNA. Now here's the nucleus. All the DNA is in the nucleus. You can see the chromatin or chromatin in here. And that's what you see over here. Here is the cell membrane. Here's the nucleus. You can see all the chromatin in there stained in blue. And these centrioles, okay, don't get those mixed up with centromere. The centrioles, there are two of them, or there's one that gets duplicated here, are sort of the anchor points on either side of the cell, on either pole of the cell, to which the microtubules are connected, which then will go over to the kinetochores, and that way you can pull the cell, will pull the sister chromatids apart. We'll look at it a little bit closer here in a second. This is interphase. DNA has been replicated, centrioles replicate, and the cell prepares for division. So preparing to divide, here's what I was just talking about. So here's one chromosome with its two sister chromatids. Chromatids are connected at the centromere, and on each chromatid has a kinetochore to which the microtubules will attach, and that way they're like little ropes. Then you have your centrioles on either end of the ropes that then pull the chromatids apart. So centrioles replicate, they're out of the picture here. Microtubules are synthesized and microtubules attach to kinetochores as the cell gets ready to divide. Now, mitosis begins a physical process of dividing. There are 5 phases here, and we'll differentiate them based on the events that take place within them. So we have prophase, prometaphase. Think about the names here, because the names of the phases will help you remember what's going on in the phases. Prometaphase, there's metaphase, anaphase, and telophase, or some people say telophase. Okay, in prophase, so this is prophase, this is before division. That's the pro part. The phase before division, I should say the phase before separation, that's pro. Chromosomes condense. Really, I should say chromatin condenses, forming visible chromosomes. Spindles form, and the nuclear envelope dissolves. It's really important to remember this. In order to get to the DNA in the nucleus, you got to get rid of the nuclear envelope. And so during prophase, the nuclear envelope dissociates or dissolves. The components remain there because it has to reform, but it dissociates. so that now there is access to the chromosomes. Pro-metaphase. Okay, so metaphase down here, meta means middle, and that'll make sense in just a second. So in pro-metaphase, this is the phase before the middle phase, pro-metaphase, where spindles attach to the kinetochores, which I showed you just a second ago, and chromosomes begin their migration to the equator. Chromosomes begin movement towards the equator. Okay, the equator is in the middle, like the equator of the Earth. It's in the middle of the Earth. That's where meta comes in. Meta means middle. So the chromosomes complete their migration to the middle of the cell. We call that the metaphase plate, or the equator. Some people refer to it as the equator. But if you run an imaginary plane through the cell, then that would be the metaphase plate. Now, in anaphase, the centromeres dissolve, right? Because you're going to pull the cells, you're going to pull the chromatids apart now. Sister chromatids are pulled to opposite poles. So ana means against. So you're going to have one set of chromatids going to one side of the cell, the other set of chromatids going to the opposite side of the cell. They're being pulled apart by those microtubules connected between the kinetochores and the centrioles, right? So they're being pulled apart. They're going to opposite sides. And then you have telophase or telophase, where the chromatids get sequestered at opposite poles. All right, that happens in anaphase, where the chromatids are pulled apart and they collect at opposite poles, and the nuclear envelopes reform. Remember I said up here we got rid of the nuclear envelope, but the components remain, because in telophase we have to reform the nuclear envelope. Now this time we're forming two nuclear envelopes because we're dividing the cell into two. So here is prophase. Now here we see chromatin in an advanced stage of condensation, because here we can start to see the very the structured chromosomes beginning to appear. So the nuclear envelope, now there's no nuclear envelope here. The nuclear envelope has dissolved, and the chromosomes, or the chromatin, condenses to form chromosomes that we can visually see with a simple light microscope. All right, so chromosomes condense and become visible. Chromosomes appear as two sister chromatids held together at the centromere. Okay, we can actually see the little X shape of the chromosomes. Cytoskeleton is disassembled. Cytoskeleton is just a structure that holds all of the components within the cytoplasm together. So that stuff has to dissolve away or disassemble, so that now we can move these components within the cell. Golgi and ER are dispersed. That's Golgi apparatus and endoplasmic reticulum. Each new cell has to have some of that stuff, and the nuclear envelope breaks down. Okay, that's prophase. This is the phase before division, prophase. In prometaphase, so this is the phase before the middle. So here you can see now very clearly, you can differentiate chromosomes or chromatids as they come apart now, as they start moving around rather. And so you'll notice that I think you can kind of get an idea here that you can draw an imaginary line through there. That's the metaphase plate. And so these chromosomes are inching to line up on the equator or on the metaphase plate. You can kind of see that, imagine that, as I tell my students. If you close one eye and squint the other, then you can imagine that's what's going on here. You can almost see it happening. Here we are in the illustration where you see chromosomes moving towards that metaphase plate. Okay, they're moving towards the middle. So chromosomes attached to microtubules at the kinetochores. Each chromosome is oriented such that kinetochores of sister chromatids are attached to microtubules. Chromosomes move to the equator of the cell. That's what's going on here. Now this is the chromosomes moving. This is before the middle, but they're moving towards the middle, as you can see here. In metaphase, they are in the middle. They are lined up on the metaphase plate or on the equator. Right? They're in the middle. That's the meta. Here you can see it. This is not a great image of metaphase, but nonetheless you can clearly see the line here representing the metaphase plate. These are centrioles right here in this little dark green. These are centrioles, and these little strands are your microtubules, which are connected to kinetochores on either side, which will then allow you to pull the chromatids apart. All chromosomes are aligned at the equator or on the metaphase plate. Chromosomes are attached to opposite poles and are under tension, right? So we're going to pull them apart. Now, here's just a quick aside on the metaphase plate. So here you can see a centriole there in the middle, centriole there. You can see clearly the microtubules, and the chromosomes are all jumbled together here, but this is the metaphase plate. This is the equator of the cell. These are called asters because they look like starbursts. And so here you can kind of barely see it. This is an imaginary plane right there cutting down through the middle of the cell. That's the metaphase plate. Now as we go into anaphase, ana means opposite. So here we're going to have the sister chromatids being pulled to opposite sides of the cell or opposite poles. And so here now the sister chromatids have the chromosomes have come apart, so we have sister chromatids going to one side and the other, but now they're no longer called chromatids, they're called chromosomes, even though it's one half of the original double-armed chromosome. When they are together at the centromere, we call them sister chromatids, but when they come apart, now we call them chromosomes. And here you can see one set of chromosomes moving to this side, the other set of chromosomes moving to that side. This would be the equator of the cell. So proteins holding centromeres of sister chromatids are degraded, freeing individual chromosomes. So the centromeres dissolve, allowing the chromatids to come apart. Now they are chromosomes, and the chromosomes are pulled to opposite poles. And then we have telophase or telophase, where we reform the nuclear membrane around this set of chromosomes and this set of chromosomes. We don't see the nuclear envelope just yet, but in the diagram you see it being reformed. There again, that would be the metaphase plate, but what is going to happen here, the cell, we're talking about animal cells now, the cell is going to pinch in on either side, sort of like if you have one of those sort of long, like a 12-inch long water balloon filled with water, and you can pinch it in the middle, right? That's kind of what's going on here. Chromosomes are clustered at opposite poles. We see them here. Nuclear envelopes reform. We see that happening here. Golgi complex and ER reform, spindles disassembled, and the cell starts to pinch in. We call that little area there the cleavage furrow, right there. We don't really see the furrow here because we don't see the plasma membrane in this micrograph, but that's what's happening in cytokinesis. All right. In animal cells, the cleavage furrow forms to divide the cell, so it's pinching in like a water balloon. Now we'll see the differences in plant cells. So this is in animal cells. Remember, plant cells have that very rigid cell wall. And so in plant cells, the cell plate forms to divide the cells in half. So the cell plate forms from the innermost point outward to form a wall between the two daughter cells, whereas the cleavage furrow forms from the outside inward to separate the two daughter cells in animal cells. Here we can see the cleavage. Now this is one big cell, but it's beginning to divide, physically divide. That's the cleavage furrow. And in fact, when we look at growth and development later on, we'll see one cell, the zygote, divide into two, divide into four, and we call those little, you can actually see them under the microscope. We call them cleavage furrows, and we call that process, in fact, when a cell is physically dividing like that, we call that cleavage. Okay, here we see in plant cells, not a really good micrograph, but you can see the cell plate forming there. But here is the plate. You can see it in green here, and it actually begins on in the innermost part and forms outward. So you can see it hasn't quite reached the sides of those cells. But so this is one big cell. Here the genome has been replicated, and the septum, the cell plate, is forming from the inside out to divide those two cells, to separate them. Counting chromosomes and chromatids. Okay, it's really important that you differentiate chromosomes from chromatids, and it can get confusing, but it need not be so. So when you have a chromosome, remember the X-shaped chromosome, like this, and it has its two sides, two arms, and they're connected at the centromere. We call these two arms sister chromatids, and they're genetically identical. Within this chromosome, they're genetically identical. Now, when you separate the sister chromatids by dissolving away that centromere, we refer to them no longer as, these are not chromatids, these are chromosomes. So that when during the S phase, replication, synthesis phase, DNA replication, these chromosomes, which are just a single arm, are replicated so that you get, I call these whole chromosomes, and that's just my terminology. It's just so I can keep it straight in my own brain. You can make up your own term, but I call these whole chromosomes. I don't refer to these as half chromosomes. These are chromosomes, but these are whole chromosomes. To me, a whole chromosome is two sister chromatids connected at the centromere. So you start out with chromosomes with a single arm, and when they replicate, then you get whole chromosomes. And then when you go into mitosis, then those whole chromosomes come apart, right? The centromere dissolves and the chromatids separate, and now they're not chromatids anymore, they're chromosomes. But in my mind, they're not whole chromosomes. But anyway, You get the idea. And so you get one set of chromosomes pulled to this pole, the other set pulled to that pole, the nuclear envelope reforms, and you have your two daughter cells. All right, so my little note here says to determine the number of chromatids, multiply the number of centromeres by 2. All right, so these are just chromosomes. There's no centromere here, so, and here the N number is 1. After synthesis, right, you have whole chromosomes, 2 whole chromosomes, and you have 1 centromere, 2 centromere. 2 times 2 is 4, so you have 1, 2, 3, 4 chromatids. 4 chromatids here, no centromeres here, so you have no chromatids. See, these are no longer chromatids. These are chromosomes. Once they separate, they're no longer chromatids. Okay, here is just an example. N equals 1. I guess I'll go through this. N equals 1 here. So 1 chromosome. So at interphase, after the S phase, you have 2 chromosomes. 2 times 2 is 4. So you have 4 chromatids, right? 2 times the number of centromeres. In prophase, still the same. In metaphase, still the same. Now in anaphase, right, the chromatids separate, so now they are chromosomes. So now instead of chromatids, in anaphase you have 4 chromosomes. No chromatids because they're all separated. And then in telophase, once the chromosomes are grouped together on either pole and the nuclear envelope reforms, then you have 2 chromosomes, right? You have 2 daughter cells, each with a chromosome. If we bump this up, now this is interesting. This really is interesting to me. If you bump this up, now this is N equals 23. What organism is that? Think about it, give you 2 seconds. Humans, Homo sapiens, right? Our N number is 23. Okay, so in interphase, this is after DNA replication, after the synthesis phase. We have 46 chromosomes, right? We have 2 sets of 23. They're identical. So we have 46 chromosomes after synthesis. 46 times 2 is 92 chromatids. Right? You have 46 whole chromosomes. 46 whole chromosomes. 46 times 2 is 92 chromatids. Remember, a whole chromosome is 2 chromatids connected at the centromere. In prophase, same thing, 46 chromosomes, whole chromosomes. That means 92 chromatids. Metaphase, same thing, 46 chromosomes. They're just lining up on the metaphase plate. 46 whole chromosomes, that's 92 chromatids. I would never ask you to differentiate like this, okay? But it's just interesting to me when we get to especially anaphase. Now remember, in anaphase, the chromatids are separated. The centromeres dissolve and the chromatids are separated, so now they're no longer called chromatids, they're called chromosomes. And look what happens here. In anaphase, before separation, there are 92 chromosomes in one cell. That to me is fascinating because remember we said before that in the first lecture that if you have more than the normal chromosome number, which is 46 for humans, then you run into problems. But here in anaphase we see 92 chromosomes. and then the 46, so you have 46 on one side, 46 on the other side, and then they separate, right? Telophase, cytokinesis happens, you get 2 daughter cells, and each daughter cell has back now 46 chromosomes. Cool? Okay, the cell cycle has 2 irreversible points. You have to be able to control the cell cycle, otherwise if something wacky happens, your DNA gets damaged or something, and there's no way to control the cycle, then that cell with damaged DNA can just replicate and replicate and duplicate and duplicate and divide and divide and divide and divide, and you get all of these cells. Now you may end up with a whole organism that has damaged DNA. So there are 2 points where you can stop the cycle, back up, regroup, and one is during the replication of genetic material, that's in the S phase, and the other is during the separation of the sister chromatids. So the cell cycle can be put on hold at specific points. We call them checkpoints, and at those checkpoints the whole process of division, of replication, is going to be checked by the cell itself for accuracy, and it can be stopped. It can be halted. You can go back and repair and then continue, or you can just kill the cell off altogether if it can't be repaired. And so these checkpoints allow cells to respond to both internal and external signals. And so those checkpoints are the gap1 S checkpoint, or the point between going from growth 1 to synthesis. It's at this point that the cell decides, I put decides in quotation marks because the cell doesn't really make a decision, right? The cell has no brain, but the natural series of events decides if that cell is going to go into division, further into division or not. And so this is the primary point for external signal influence. In other words, if you're going to have something go wrong to the cell due to some external event or external substance, it's going to be at this point. Now the gap2 or growth 2 / M, that's the mitosis checkpoint, is where the cell, we finished up synthesis, we're into gap2, we're getting ready to go into the physical division, mitosis. There's a checkpoint right there. And here's where the cell makes a commitment to physically divide. The cell checks the success of replication because we've just come out of synthesis. And so if the DNA was not replicated quite right, if there's something wrong, if there's a mistake in the DNA that could not be fixed, then the cell can take care of it at this point before cell division. You want to make sure that you take care of these problems before you duplicate the problem, right? And then within the M phase, the late metaphase, we call it the spindle checkpoint. And here's where the cell ensures that all chromosomes are attached to spindles, because remember, it's here that the chromatids are separated and you actually divide into 2 daughter cells. And so if something is wrong there, if somebody's not connected, right, that's where you get a cell with 45 chromosomes instead of 46, right, when things don't separate properly. So we call that the spindle checkpoint. And we see them here in this diagram. Okay, here's the regular cell cycle, the diagram that you're familiar with. There's G1, S, G2, and here's the mitosis phase, the division phase, and there's cytokinesis. So there's a checkpoint called the G1S checkpoint here before the cell goes into synthesis, right? So if something is going on There's some external component that is affecting the health of the cell. We have a checkpoint here. We're going into synthesis, so we're duplicating, replicating DNA. Here's G2, and here's the G2M phase. So, okay, so the G1S phase is when the cell decides that it's safe to go into synthesis. The G2M phase is when the cell decides, okay, it's safe now to divide. In other words, all the DNA has been checked over. All the DNA is either correct or mistakes have been found and they've been corrected, so now we can move on into division. And at the spindle checkpoint, that is when the cell determines that all chromosomes are connected at their kinetochores by the spindles, by the microtubules, so that once physical separation takes place, everybody goes to their proper place. If not, then the cell can shut the process down. Okay, again from Professor Dave, the cell cycle and its regulation. So this is a good little simple video that's going to show you the cell cycle and how the cell cycle is regulated at those various checkpoints. And finally, we want to wrap this up by talking very briefly about cancer. Cancer is unrestrained, uncontrolled cell growth, and it's a failure of the cell cycle control. So remember, we have those three checkpoints, right? G1S, G2M, and spindle. And if something goes wrong at any one of those, we can fix it. Or if not, if something happens, something flaky happens and a problem does not get fixed, then the cell can go into uncontrolled cell division and growth, and that's how you end up with cancer. So there are genes called proto-oncogenes. Now, onco means cancer, so these are genes that cause cancer, but these are protogenes, so they're not cancer genes, but they could potentially cause cancer or turn into cancerous cells. So these are normal genes, but when damaged, they accelerate cell growth. So here we see in the little diagram over here, oncogenes and tumor suppressor genes. Here's the normal cell. That's a proto-oncogene. And if something happens, right, then we get that cell turn into an oncogene or a cancer gene that then goes into uncontrolled growth and division. We call those tumors. Now here tumor suppressor genes. Tumor suppressor genes, and we'll talk about P53 in a second, but tumor suppressor genes suppress cell growth. So their job is to, if there is a proto-oncogene that for whatever reason is trying to go into an oncogene, tumor suppressor gene can stop that, can halt it, so that you do not have growth of a tumor. Tumor suppressor suppresses the growth of tumors. However, when they are damaged, then cell growth continues unchecked, and now you have cells that are uncontrollably growing and dividing, and we call that a tumor. I call this P53, I call it the master gene, because it is the #1 gene that sort of controls whether a proto-oncogene will go into an oncogene. So normal P53, it's a protein, it's a gene, it's a protein called P53, and it says normal P53, it identifies DNA damage or cell cycle abnormalities or hypoxia. Hypoxia is a lack of oxygen, reduced oxygen. Of course, we need oxygen in order to survive. So if any of these things occur, P53 goes into action. Now it can do one of two things. It can arrest the cell cycle and initiate DNA repair, and if DNA repair is successful, then the cell cycle can get restarted. If P53 notices any of these things, it will arrest the cell cycle, and if the DNA cannot be repaired, then it forces the cell into, we call this apoptosis. That's programmed cell death. That's the death of the cell. That's the death of a broken cell. We're going to get rid of the broken cell so that it can no longer cause any damage, or it can cause no damage in the life of the organism. If any of these things are noticed, P53 can go this route, or it can send a cell directly to apoptosis. But it's going to try to correct anything that it can. So it'll stop the cycle, repair it if it can. If it can't repair, then apoptosis. Now, problem here is with P53, if the P53 gene becomes mutated or it itself becomes broken, then these things get noticed by P53, but here's the little broken P53. P53 can no longer do anything. It can no longer repair. It can no longer send a cell into apoptosis because it itself is broken. It can recognize these things, but it can't do anything about them. And so what happens is, because this can't do its job to check, then the cell, the broken cell, the damaged cell, continues into the cell cycle, continues to duplicate and replicate and divide, and then that cell divides, right? One divides into 2, divides into 4, divides into 8, divides into 16, and so on and so on and so on. And those groups of cells, those bunches of cells, can become tumors. All right, that's the P53 master gene. If you have any questions, you can message me, 804-229-7993. Email me, jjeffries@rbc.edu. You can also message me here on your inbox and let me know what's going on. Thank you.